Olá, boa tarde. Retornamos, então, ao simpósio Inteligência Artificial para a Conservação da Biodiversidade, e hoje de tarde teremos, então, a nossa última sessão com a presença do pesquisador Dr. Barnaby Walker. Vou apresentá-lo rapidamente. Barnaby é pesquisador do Royal Botanic Gardens, Kew, no Reino Unido, onde desenvolve pesquisas sobre o aprendizado de máquina para avançar nos estudos em biodiversidade e conservação da natureza. Sua pesquisa atual se concentra na construção de redes neurais para explorar novas maneiras de interagir com imagens de espécimes do herbário Kew, com o objetivo de encontrar possibilidades para seu uso direto em pesquisas. Sua função anterior como analista de ciências da conservação envolveu o uso de modelos de aprendizado de máquina para prever o risco de extinção de espécies de plantas e orientar as prioridades no processo de avaliação do risco de extinção. Enquanto trabalhava na equipe de avaliação e análise de conservação do Kew, ele também ajudou a construir a ferramenta Rapid List Concern, um aplicativo da web usado para gerar rapidamente avaliações da lista vermelha da IUCN para espécies de plantas de baixo risco de extinção. A palestra do Dr. Barnaby será em inglês e vocês podem participar, né, trazendo perguntas através do nosso chat, tanto em português quanto em inglês. As perguntas em português eu poderei traduzir, né, ao final da apresentação. So, welcome, Dr. Barnaby Walker. It's a great pleasure to have you here participating in our conference as a speaker. So, thank you in advance for accepting talking for us, and have a good presentation.
Thank you, Joel, and thank you for inviting me. Thank you to everyone listening, and I'm sorry that I can't speak Portuguese well enough to present in it. But so, as Joel said, I'm a research fellow at Kew Gardens, and I'll be talking about some work that I carried out in the conservation assessment and analysis team there on using machine learning to guide our extinction risk priorities. And so, first of all, why am I talking about extinction risk? Assessing extinction risk? Well, I think Patricia from CNC Flora gave a very good introduction explanation of how we assess extinction risk yesterday. But the reasons we think it's important at Kew is that when we've got limited conservation resources worldwide, finding out which species are threatened is really important to allocating those resources well. And it's also really key to understanding the drivers of biodiversity loss and hopefully preventing it. And so at Kew, we focus on the IUCN Red List because it's the most authoritative source for global extinction risk assessments. And just a little refresher on what the Red List is. So for people that aren't familiar with it, it's a set of quantitative criteria that are used to assign species, whatever taxa they are, from plants, mammals, birds, mollusks, assign one of these extinction risk categories to them. And it's really important to know that the Red List isn't just a list of the threatened species, it's a list of every species that has an extinction risk assessment. So the extinction risk categories actually go from least concern, which is the least threatened category, up to near threatened, through the threatened categories, vulnerable, endangered, and critically endangered, and then onto the extinct categories, so extinct in the wild and extinct. And if a species doesn't have enough information to carry out a proper extinction risk assessment, then it's just given a data deficient flag. And throughout my talk, when I get onto our uses of AI, I'll be talking about predicting whether species are threatened or not threatened. And so that's really just focusing on predicting whether a species is in one of those threatened categories, so critically endangered, endangered, and vulnerable. And so, as well as being the most comprehensive source of global extinction risk assessments, at Kew we think the IUCN Red List is really important because it's also a comprehensive documentation of our current state of knowledge about the threats to the species on it. And for that reason, it can form the basis for protecting areas. So at Kew, we've got another program called the Tropically Important Plant Areas, and the extinction risk assessments that our plant assessment unit does feed into that process and feed into form some of the basis of designating areas as important plant areas. But the Red List assessments also contribute to policy decisions. So recently, you might have seen in the news that the president of Cameroon revoked logging concessions in Igbo forest, and some of the evidence that formed the basis of that decision was extinction risk assessments carried out in our plant assessment unit. However, we've got a long way to go if we want to assess all the status of all plants. So currently, just over 10% of all plants have a global Red List assessment. But that doesn't show us the full picture of how far we need to go, because that 10% has a lot of gaps and biases in it based on the criteria that people use to choose which groups to assess. So here we can see some of the geographic bias. So on the left, I've made a map showing the species richness in each botanical country, just taken from Kew's World Checklist of Vascular Plants. And then on the right, I've made a map of the proportion of each botanical country that has a global Red List assessment. We can see some real discrepancies here. So some of the centers of plant diversity, so South America, Southeast Asia, some of those areas have the lowest proportion of species assessed, while some areas that aren't particularly species rich, like parts of Northern America, have quite a large amount of their flora already assessed. So we can see some geographic biases there. We can look at this in a more systematic way if we ask how representative the Red List is as a sample of all vascular plant diversity. And on this slide, what we've got is a comparison between plants on the World Checklist of Vascular Plants, so that's all vascular plant diversity, compared to those that have been assessed for the IUCN Red List, split up by different attributes. So here, we're looking at whether a species is endemic or not to a botanical country. And we can see that endemic species make up about 56% of the World Checklist of Vascular Plants, but they only make up around 49% of species that have been assessed for the Red List. So that shows us that there's a slight underrepresentation of endemic species on the Red List. And then if we look at species with a recorded human use, we can see the opposite picture. So species with a recorded human use are overrepresented on the Red List. They make up about 25% of it, while they only make up about 10% of the World Checklist of Vascular Plants. If we look at what continent vascular plant species are from, the sample looks a bit more representative, but we can still see that species from Africa are overrepresented, while species from Southern America, tropical and temperate Asia are slightly underrepresented. And then if we look at the life form of species, we can see that woody perennials, so trees and shrubs, are vastly overrepresented on the World Checklist, while all other life forms are underrepresented. And then finally, if we look at family here, I've just ranked these from top to bottom with the most underrepresented at the top, the most overrepresented at the top and the most underrepresented at the bottom. And we can see if we look at the top, the three most overrepresented families are cactuses, Cactaceae, legumes, Fabaceae, and the myrtles, Myrtaceae. And then if we look at the bottom, the four most underrepresented families are the daisies, Asteraceae, orchids, Orchidaceae, grasses, the Poaceae, and the mint family, Lamiaceae. And what's really striking about this is that these four families make up a huge amount of vascular plant diversity, about 25% of all vascular plants, but they only make up a small fraction of the Red List. So we can see that there are really quite big gaps in what we have assessed for the Red List. And that leads us on to the question of how can we start to fill in these gaps? And as I go on, I'll show you how Kew has been trying to answer this question by using automation, machine learning, and statistical modeling. But it's really important to acknowledge that these automated techniques, this AI approaches, have really been facilitated in part in recent years by just a vast amount of data that's been made digitally available. So things like specimen occurrences through big aggregators like GBIF and BN databases, but also resources provided by different countries. So I've just got a picture of species link here provided by Brazil, which feeds into some of these aggregators, but is also a very rich source of information in its own right. And then also databases of taxonomy, like Keith's World Checklist of Vascular Plants, and other trait databases. So without these digitally available data, it would really make using automation or AI impractical for trying to fill in the red list. And so there are two ways that I'll focus on how we've been using it at Kew. The first is using AI to get a broad view of the red list of what's threatened and not threatened. So to get a broad view, in our State of the World's Plants and Fungi report, we did analysis where we corrected the IUCN sample by using a technique called multi-level regression and post-stratification, which is a statistical modeling technique that's been used quite a lot recently in forecasting political polls. And what it allows you to do is to take an unrepresentative sample, like an opinion poll, or in our case, the IUCN Red List, and apply a multi-level regression model to different attributes that we think influence that. So for us, it's those attributes I went through, looking at how representative or unrepresentative the IUCN Red List is. For political polling, it's often municipality, age, level of educational attainment. So you apply your multi-level regression to the sample that's been split into those groups to get an estimate of the mean value that you're interested in for each of those groups. And then you scale up that model using post-stratification, which is basically taking the numbers for your population of interest. So in our case, that's all vascular plant diversity. So you take the population that's in each of those subgroups and scale up your estimate from your model. And if you look on the right here, these are some of the results that we got. So, well, the top value is the result that we got. So the blue bar, faded blue bar, is the uncertainty on our estimate. So the stronger the blue color, the more certain we are that our estimate falls in that area. And the circle is the median estimate. So using our MRP model, we estimated that 39% of all vascular plants are threatened. And that's a bit lower than the value you'd get if you just took the estimate from the raw numbers from the IUCN Red List. But more strikingly, it's quite a bit higher than a previous estimate made in 2016, where they just took a random sample of representative plant groups. But another interesting way that we can use this model, rather than just getting this overall picture, is we can split up our estimate by the subgroups that we applied our model to. So here I've split up our estimate by family. And again, the blue bars, the uncertainty on our estimate. So the stronger the blue color, the more certain we are that our value falls in that area. The open circles are the median estimate for each family, and the closed circles are the estimate for each family that you'd get if you just took the numbers straight from the red list. And what's really striking is you can see Myrtaceae up here at the top. Our estimate from our model comes out making Myrtaceae a lot more threatened than it would be if you just looked at the red list. So in that case, we're saying that our model is indicating that Myrtaceae, the level of threat for Myrtaceae, is underestimated on the red list. Whereas if we look on the bottom, we can say that the level of threat for Ebonaceae is possibly overestimated on the red list. And it's really striking in the case of Myrtaceae because this was one of the families that were overrepresented on the red list. So we've assessed a good portion of the family, but that's been heavily biased to species that are less threatened. And as we assess more and more of the family, we should see the level of threat for Myrtaceae going up. But it should be said that these uncertainties on these families are quite high. And that uncertainty really depends on getting the right choice of attributes to feed into our model. So at the moment, we don't think we've got the choice of attributes quite right. We need to work on our model a bit more. But we can already see that this is quite a promising technique for identifying over and underestimated groups and trying to guide which groups we should assess next. So then we can look at, so that's taking a broad view of filling in the gaps in the red list. We're also looking at filling in gaps by estimating the threat status of each individual species on the red list using AI. Each individual plant species worldwide using AI. And there's two ways that people have done this in the literature. The first is to calculate metrics that are typically used in the assessment process and use predetermined thresholds on those to classify species as possibly threatened and possibly not threatened. So these are more automation than artificial intelligence. And two examples of these, the first that I've got on the right here is an example from last year where Steva et al. Used these predetermined threshold method to generate preliminary assessments for the flora of tropical Africa. And then on the bottom here, we've got a screenshot of the Rapid Least Concern app that I helped develop while I was in the Conservation Assessment and Analysis team, which we used to generate documentation for species that we could be fairly sure would come out of the assessment process as least concern. But the second approach to estimating the threat status of each individual species is to learn these thresholds from past assessments using machine learning. So two recent examples. The first is Pelletier et al., where they used machine learning to predict plant conservation priorities globally, so which areas of the world we should focus conservation priorities on. And the second is a more recent example where Alexander Zizka and co-authors used deep learning to generate preliminary assessments for the whole orchid family. And so before I get onto how we've been using these at Kew, I should say that most of these methods are primarily based on occurrence records. So the occurrence records are the primary data source, and they're usually taken from herbarium specimens like this one that I've got on my slide, but more and more in places like GBIF, aggregators like GBIF, citizen science observations, so things from iNaturalist or plot observations are being fed in as well. And the automated methods typically calculate measures based on these occurrence records, so range as extent of occurrence or area of occupancy, threat level based from rasters of things like human population density or human footprint index, as well as environmental variables and things like the number of occurrence records. And they use their predetermined thresholds on these values. And so at Kew, like I said before, we've developed this Rapid Least Concern app, which falls into this class of predetermined threshold methods. And if it will work, actually, no, I don't think it will work. I was going to give you a little quick tour of it, but if you follow this URL, you can go and have a look yourself. And I think it's quite simple to use. You just type in the name of the species, you select the right taxonomy from the right-hand side, and then you click Run Analysis, and it will give you a map of where the species is. It will tell you whether it's probably least concern or not, and it also generates the tables that you need to submit that least concern assessment to the IUCN. But possibly more interestingly, we're also using the AI technique to predict which species are threatened and which aren't. But we're not using the AI techniques to submit assessments to the Red List. Firstly, because the threshold of evidence that you need to submit a threatened assessment to the Red List is quite high, so they probably wouldn't accept them. But also because we can't really be sure that each individual prediction is 100% correct, and the stakes are really high. So for us at Kew, we're using our machine learning predictions to guide our assessment process. So here on my slide, I've got predictions that we've made from a random forest model, going from the predicted least threatened at 0 on the left up to the predicted most threatened. And these species are in a group. There are all legume species from the Caesalpinii group. And there's two ways that we can use these to guide our assessment process. So the first is to prioritize those species that we think should be assessed next by which we've predicted to be most threatened. So you can see the 10 most threatened here, and we prioritize those for assessment first. Or the second way that we can use these to guide our assessment process is to look at the most uncertain predictions. So these error bars on our predictions show us how uncertain the model is in the prediction. And here I've just highlighted the 10 most uncertain, and we'd take away those 10 species and we'd focus data collection on those species. So we'd collect more data, clean more occurrence records to try and get high quality data to then feed back into our predictions. However, it should be said that we need to use these automated methods and AI methods quite cautiously. When we're reporting the performance of our methods, it's really important that you don't just report one metric. So typically people just report the accuracy of the method. It's really important to split that up into how well your method performs on threatened species, how well it performs at predicting not threatened species, so you can give a clear idea to the users how your method will perform in each case. And we also really need to weigh up the limitations of the data. So different groups will have data that's in different states. So some groups of species will have less data, some groups of species will have dirtier data, just based on the history of collections of them. And so when we're developing these techniques, we really need to test them on multiple groups so we can weigh up the limitations of them and find the right technique for the right problem. And then finally, one thing that really helps with these is explanations of the predictions. So for our machine learning random forest example before, we can get explanations for these predictions in a few ways. So one is the overall explanation of which of the predictors that we put into our model are most important for our predictions. And we can see here from this plot, for our model, the range was the most important predictor, followed by human population density, and then followed by the rest of the predictors. So we know that our model is making predictions primarily based off of the range and the human population density, which really, for us, is reassuring, because that's typically what the human assessments go off of. And then secondly, we can also get individual explanations for each prediction. So here we've got a depiction of a technique called SHAP, which is a game theory method where each prediction is split up into the contribution of each predictor. So you get the average prediction, and each predictor pushes that prediction up away from the average prediction or down below the average prediction. So for this species, Mercium macaca, we can see that the low EOO, range size of 0, is pushing that prediction up to more threatened levels, whereas the very low human population density is pushing it back down. So this prediction of 0.2 would actually classify our species as not threatened, and that would be based on the low human population density. And the good thing about this explanation is that it lines up very well with the assessment justification on the red list for this species. So this species is actually assessed as least concern, because even though it's got a very low extent of occurrence, it's in an area where people typically don't live, so there's no threats for it. And so these explanations can help us to sense check our methods. And with that, hopefully I've given you a good overview of how Q is using artificial intelligence and automation to guide our assessment processes. Thank you for listening.
Thank you, Bess, for the great talk. Very interesting. And we could see a bit of the methods you guys are using to advance in this artificial intelligence to guide conservation actions. So for now, I will remove your presentation and ask Cristiano to get back to discuss with us. And then I will ask the public for questions if they have. And, bom, agradeci a participação do pesquisador Barnaby Walker e abro agora, então, para discussão. Enquanto eu recebo as perguntas de vocês aqui no chat, eu vou fazer algumas perguntas que eu anotei durante a palestra. So, Barnaby, I will start the questions. So for many species that have like small populations restricted to small patches, they are very threatened, because if you have like one event of extinction in that place, we will lose the entire species. And for large countries like Brazil, we have many small collections countryside, and many times these samples from rare species are not in the big area, but just in small collections. So these collections many times also are not digitalized and are not available for producing records to analyze in conservation assessments. So how could we deal with that, and how can we improve the data for species distribution records considering these small collections?
Yeah, that's a really good question, because something we found applying our machine learning methods is that because we typically use data from GBIF, so one of the big occurrence record aggregators, is we find that we can't make predictions for a lot of species just because there's not the occurrence records available for them. And if we were able to access these small herbaria, then I think it would really, really increase the reach of these methods. But I think, so you mentioned that one problem is resources. So they typically don't have the resources to digitize, or they just haven't digitized them. But also I think another tension is attribution for these herbaria. So I know GBIF have done a lot of work on getting people to cite DOIs that can then be used for tracing metrics for herbaria or other institutions that contribute data. But I think for these smaller herbaria, it's really hard to track that. And I think one thing that maybe might help is if we were able to attribute in research when we use specimens from different institutions, if we were able to give attribution to them in a more transparent way, that might help with showing the importance of them. Because once we show the importance of these collections, hopefully we can get more resources to them for digitizing. I think also maybe larger institutions could possibly do more with resource sharing. So as well as money, also knowledge as well on how to run the digitization. So
Yeah, it is happening somehow here in Brazil, since we had the Reflora program, which is led by Rio de Janeiro Botanical Garden. They have like stations for photographing the plants and digitizing the data, and they are sharing these stations with small collections countryside. So this is a good way also to improve the data online.
Yes, I really like the Reflora project. I think Q was a partner on it before I joined, but we put out a paper a year or two ago in collaboration with colleagues at Rio de Janeiro, that showed the importance of the online resources that came from Reflora as well. So I think projects like that are really important and really effective.
Yes. I have another question. Do you have any idea, considering the species that you have worked already, what's the percentage of species with deficient data?
Yes, I could probably look this up on the Red List on their website. You can get up little graphs. Off the top of my head, I can't remember. It's probably between 10 and 20%. It's relatively high, but a lot of work goes. I know the assessors, when they do the assessments, they do a lot of work to try and avoid giving something a data deficient flag. So it's sort of a last resort. But there has been work done on well, so for our machine learning techniques, we typically treat the data deficient ones as unassessed, and we generate predictions for them as well. So as well as addressing things that haven't been assessed, these methods can be used to make up for data deficient things as well.
And I have one last question before presenting the questions from the public. This morning, we were talking about conservation on animals. And so during your talk now, I was thinking about plants and animals that co-evolved and are completely dependent one of another. And I have never seen any publication talking about that when considering conservation assessments. Have you seen anything, or do you know any project in this area?
I'm not sure about red list assessments specifically, but I know there's been work on things like butterflies. So because the migratory patterns of butterflies are changing because of the availability of plants flowering at the right time, it's really affecting the sort of extinction risk of these species. So I think it is factored into the red list assessments for things like the pollinators. And sometimes you'll see on assessments that a justification for something being assessed as threatened is that the pollinator for that particular plant is disappearing. But it's not as common because the evidence isn't as strong. So I think there are projects. I can't name any off the top of my head, but I think it's definitely being looked at for butterflies.
Ok, thank you. Cristiano, queres falar alguma coisa, perguntar alguma coisa?
Hello, I have one question. Thank you. Thanks very much, Barnaby, for your presentation. It's a very interesting work. What's the knowledge, or actually recent knowledge about plant population rates, mainly in the tropics? I work with insects, and for butterflies, it's still incipient to use in IUCN Red List, and then we work with occurrence data only. And for plants, it's a good knowledge about population rates.
It's a similar problem for plants. So some species that are of particular interest to people will know about the population sizes, but it's missing for a large amount of plants. So most assessments are only done off of occurrence records, which is a shame because that population data is really, really useful. So hopefully it will be recorded more and more. I know at Kew, we're trying to get, when people collect seeds, to put an estimate of the population size, but again, it's a difficult thing to record as well. So yeah, we're in a similar situation.
Yes, thanks.
Okay, you have some questions from the public. So the first one is from Luis Fernando Esser. Is Kew thinking in improve these methods to apply in red lists of ecosystems as well?
I can't think of any work. Definitely there isn't any work in the conservation assessment and analysis team at Kew going on for that, but there is interest in doing that. So at Kew, we're in the process of developing our science strategy for the next 5 years, and I hope there'll be something about applying these methods to the rest of ecosystems in there. Even if there isn't, I think there's interest to start doing this, but nothing at the moment.
Okay. And we have another one from Renato Ramos da Silva. How to efficiently aggregate databases from different sources? What are the most sensitive aspects?
Yeah, so I think the biggest problem with aggregating databases from different sources, at least for occurrence records, is duplicated records. Because just when you're collecting plant material, when people collect them, they collect duplicates, they're sent to different herbaria. So the databases for different herbaria will have the same record, but there's often a lot of missing data. So if it's an old specimen, there might not have been much written on the specimen sheet, or it might just have got lost in the digitization process. So matching those duplicates, I think, is the trickiest thing. And there's a lot of work done on cleaning your data sets after you've aggregated them. But yeah, I think matching those records, as well as the taxonomy as well. Sorry, different taxonomies are different to match because those duplicates might have gone to different places. Someone might have changed the determination on one, and it might not have been changed in a different institute. So then you've got things that might not look like duplicates but are, and then you have to resolve that taxonomy as well. So duplicates and aligning taxonomies, I think, are the hardest things.
And yesterday we had a talk from Patrícia da Rosa, who works at the National Conservation Center, Centro Nacional de Conservação da Flora in Rio de Janeiro. And she was telling us about how long it takes to do all the process of assessments. And I was wondering about, if you have to choose, how could you do, or what do you do first to accelerate the process? Focus on least concerned species and try to assess a large number of species, or focus on that species that you think is rare, but you don't have enough data, and then you have to go to field work to collect the plant, and then to be able to assess for conservation stats?
Yeah, so I think there's well, my ideal would be both, but if I had to choose, I'm not sure. So there's a tension in that between the different uses of the Red List. So I think there's a tension between using it as a barometer of life, so using it to get a sense of how threatened things are and trends in how threatened things are, against trying to get the most threatened things listed quickly so that that can be used as the basis for protecting them. And I think if I had to choose between the two, personally, I think it's more pressing to get the assessments for the more threatened things, so that we can, because the Red List is often used as a basis for policy, basis for protecting areas, so that we can get a sense of where these threatened things are quickly. I think I'd choose that, but if that was our choice, we would have to take into account that we then couldn't use it as a good judge of how threatened things were.
Yeah. And we have another question from Luis Fernando Essa. I know you guys work really close together to IUCN, so he is asking if you guys, how is this process of working together with IUCN? How does it work for you guys at Q?
Yeah, so I am. Well, so I'm not in the conservation assessment and analysis team anymore, but when I was slightly separate from the team that worked with the IUCN. So our team that do assessments is called the Plant Assessment Unit, and there, I think the funding for permanent members of staff. No, so I don't think I can answer questions about how the funding from IUCN comes, because I don't know, because I'm a bit separate from that. I do know that they were in communication with the IUCN a lot, and the IUCN offered training for assessors, or training for people to then become trainers for assessors. So I think it's quite a close working relationship, but I never worked directly with the IUCN because I wasn't in that assessment unit team.
But we have a very straight collaboration with IUCN on the trainings, and also producing the red lists, or the assessments to include in the red lists. Yeah,
so all of the assessments that we do at Q are for the IUCN. They all go to the IUCN Red List, and contribute to that. and all of the training that we do is training people to do IUCN Red List assessments.
All right.
Sorry, I can't answer that more fully.
Yeah. I guess we don't have more questions, or if you guys watching are you have just last chance to send one question, or meanwhile, we would like to ask Barnaby Walker again for your great talk and to be part of our conference. And it was a great pleasure to have you here.
Thank you again for having me, and thank you for the chance to speak to everyone.
Thank you. Cristiano?
Thank you very much, Barnaby, who stay with us and show your very interesting project.
Thank you.
Thank you, Bess. And so now I will end this section, and then Cristiano and I will do the closing section of the conference. So thank you, Barnaby. See you. Bom, chegamos ao fim, então, do simpósio Inteligência Artificial para a Conservação da Biodiversidade. Foi uma experiência muito bacana, assim, participar e organizar esse simpósio junto com o Cristiano, e eu acho que nós tivemos um resultado bastante positivo, tivemos mais de 300 participantes, como eu falei na abertura, né, participantes de diferentes instituições, diferentes estados brasileiros, e também de alguns outros países da América do Sul e do Reino Unido, né? Então, tivemos, assim, superamos a expectativa, eu poderia dizer, e agradeço a presença de todos, a participação bastante efetiva nas discussões, nos chats, né? E espero que tenham também desfrutado do evento. Professor Cristiano?
Então, João, eu queria agradecer. Chegando agora, né? Portaria da gestão nova recém saiu. E estou muito feliz de estar aqui, ter organizado contigo esse evento. Acho que é importante, né? Visto que o CNPq está numa penúria aí, mesmo assim, valorizando essa Semana, né, Nacional de Ciência e Tecnologia. E aí veio bem de encontro ao que a gente tem feito, né? E tomara que a gente consiga manter o público aí ativo e atuante. Quem sabe a gente não propõe outros eventos mais para frente, né? Então, queria agradecer a presença de todo mundo. A gente eu acho que realmente superou nossas expectativas e foi um evento muito legal. Está disponível no YouTube, né? E quanto mais divulgar, melhor. Então, fiquei muito feliz de ter participado. Apesar de ser tão pouco tempo, foi bastante intenso, né? E foi muito legal. Então, queria agradecer mais uma vez também a organização, a tua organização, né, João, que puxou tudo e fizemos as coisas acontecer em tão pouco tempo, de uma forma tão legal. Eu acho que era isso. Esperamos repetir.
Obrigado, Cristiano, por todo o apoio, toda a ajuda que tu deste também na organização. Acho que é por aí, a gente vai trabalhando em time, né, e vai construindo juntos. Então, o Cristiano acabou de ser eleito vice-diretor do Museu de Ciências Naturais Carlos Ritter. Eu fui reeleito o diretor, então a gente inicia agora a gestão 2018-2020. 2018 ainda ano de comemoração do cinquentenário do nosso museu. E, claro, a gente está com as portas fechadas, né, por conta da tentativa de contenção da pandemia, mas também uma oportunidade muito interessante para a gente usar essas ferramentas que já estavam disponíveis há tanto tempo, né, de recursos online, né, como a organização desse evento, que nos permite ter a participação de palestrantes de fora da nossa região e também a participação do público, né, que vem de tantos lugares. Então, é uma experiência muito boa. Eu acho que é uma coisa, assim, que a gente precisa fortalecer daqui para frente, né, e adotar, né, para as nossas instituições. Então, eu acho que foi uma oportunidade de aprendizado e agora que a gente faça o melhor proveito, né, dessa situação. Então, muito obrigado mais uma vez a todos os participantes e até logo.
Até logo, gente.
Tchau, tchau.
Tchau.